Good afternoon. I'm Dr. Brian Shapiro, a Scientific Content Specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Organoid Culture Fundamentals, Critical Steps for Success, presented by Mr. Steve Budd. Dr. James Clinton and Himanshi Desai appear in the tutorial segment of this presentation. Dr. Clinton then joins us as a panelist during the Q&A session. Mr. Budd is a product line business specialist at ATCC, while Dr. Clinton is a senior scientist. In this presentation, Mr. Budd will give some background on the Human Cancer Models Initiative, describing organoids and other advanced models. Then, he and Dr. Clinton will tap into ATCC's vast cell culture experience and share the best practices for culturing organoids that ensure optimal results and performance. After he closes, Mr. Budd and Dr. Clinton will join us for a lively Q&A session that addresses your most technical challenges in organoid growth. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I would like to welcome Steve Budd.
Thanks for the introduction, Brian. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and Services Center in Gaithersburg, Maryland. We're an innovative R&D company featuring gene editing, differentiated stem cells, advanced models such as organoids, which is going to be the topic of today's presentation. During this presentation, we will give an overview of the Human Cancer Model Initiative and give descriptions of the models we have. We'll show what support is offered for these models in the way of clinical and genomic information, as well as organoid culture support. And we're going to play a 20-minute organoid culture video that will thoroughly demonstrate these culture techniques. So you definitely want to stay for that towards the end. So there's an unmet need for better preclinical cancer models. And why are existing cell lines used for preclinical studies insufficient? So many models that are available are in the way of classical cell lines, but often they're lacking specific tissues such as liver and pancreas that are not very readily available. These models are unrepresented in research and in clinical studies. Many cell lines lack historical data, both in the way of patient demographics and clinical outcome. Also, many cell lines may have been in culture for so long, it can be hard to determine how representative they are to the original models. Current cell line models, consisting of single cells, do not imitate the complexity of cancer diseases. They lack the genetic diversity and different cell types that exist in the real cancer tumors. So in short, cancer cell lines are not true biological representatives of tumors. They lack biological relevance. More advanced models, such as patient-derived organoids, can help address these deficiencies. As an overview of the HCMI program, the key players are divided between the founders and model developers. The founders who fund the program include the National Cancer Institute, Cancer Research UK, the Hubric Organoid Technology Foundation, and the Wellcome Sanger Institute. The model developers are the ones that actually generate the models from the original patient-derived biopsies, and they consist of the Broad Institute, Cold Spring Harbor Laboratory, Wellcome Sanger Institute, the Hubric Organoid Technology Foundation, University of Verona, the Hubric Institute, Stanford University, and Weill Cornell Medical College. ATCC was selected to be the sole distributor for the models. In the next slide, I'm going to go into more detail about how this program works. This is a workflow that describes how the models are generated and distributed. The model developers work with clinical sites where biopsies and clinical data is taken. Biopsies will be sent for both sequencing and to the model developers for generations of the in vitro models. The models themselves will be sequenced and sent to ATCC for distribution. A subset of models that have been expanded to ATCC will be sent back for resequencing. ATCC will then continue to expand and quality control test the models before distribution to the biomedical community. One important aspect of the uniqueness of these models is the level of characterization they undergo. This includes molecular and clinical data. The desire is to make available whole genome sequencing and whole exome sequencing for all of the models. This sequencing is done for the original biopsies and the further developed models as well. A significant amount of patient data is also collected, which includes diagnosis, patient background demographics, and the treatment the patients received, as well as the resultant outcome. A little later in the presentation, I'll show you in more detail how this data can be accessed. The HCMI models consist of different technologies. This presentation is going to focus on organoids and organoid cell culture techniques. But in addition, we do have various other model types, such as conditionally reprogrammed cells, a large assortment of neurospheres, which are these 3D spheroid-type glioblastoma models, and other more standard 2D models that may represent more rare cancer types. So specifically, what is an HCMI organoid? Note, organoids can be made in various ways, such as through normal tissue or iPSCs. So organoids in general are self-organizing microtissues that grow in a 3D extracellular matrix. These HCMI models are unique. As I previously mentioned, they are all patient-derived from biopsied tissue. They consist of multiple cell types and exhibit cellular polarization. This allows for in vivo architectural features. As an example, in this image, you can see the lumen formed, which is a common feature for organoid models. And similar to classic cell lines, they exhibit long-term expansion, so they can undergo multiple passages while being shown to be phenotypically and genetically stable. Here I'm going to touch on the organoid culture technology, which will be the focus of the video you'll shortly see. The technology originally came from Hans Clever's lab at the Utrecht Institute in the Netherlands. As it applies to human organoids, it relies on advanced medium that can include conditioned media from Wnt or spondin cell lines, recombinant proteins, and small molecules. This diagram is an overview of the process. So to begin, suspended cells and fragments are dropped into the extracellular matrix. They are incubated at 37 degrees, where the matrix polymerizes into a gel, forming a dome structure. The liquid medium is overlaid on the dome. The cell structure is then developed and enlarged into complete organoids. The bright field image on the right is an example of a real model, so you can see what it actually looks like. One thing to note is that it appears to be a ring-like structure due to being an optical cross-section, but it's really more like a structure like a balloon. That's a better way to sort of imagine it. To passage the models, the organoids in ECM is collected, the ECM is removed, and the organoids are enzymatically disassociated. At this point, they can be replated and expanded again. For the next few slides, I'll focus on the resources available to access supporting information, such as the clinical and sequencing data, as well as information and support that ATCC offers. Here we have the HCMI searchable catalog managed by NCI. It integrates some clinical information and allows you to search for the model based on characteristics of your choice. You can search for models of interest based on a number of filters such as primary tumor site, model type, diagnosis, stage, grade, and histological type, as well as gender, age, and ethnicity. One thing to emphasize is that it links directly to the ATCC product page where the models can be purchased. So if you go to this catalog and narrow your search until you find the model you want, you can be directed to its location on ATCC's website. The Genomic Data Commons, or GDC, is another NCI-managed site for a number of cancer data sets. This is not HCMI specific, so to find HCMI models, search for the HCMI CMDC project to find the list of models from the HCMI. Here you can download the aligned reads from the whole genome and exome sequencing and give gene expressions from the RNA-seq data. ATCC's website delivers information about the models that are available for purchase. It gives overviews of the HCMI program and emphasizes actual cell culture techniques and information. Each individual model page will give guidance on the exact complete medium formulation specific to a select model, as well as subculturing, thawing, and freezing protocols. Protocols exist in various formats, including individual documents on each product page, an organoid cell culture guide, and a publication on organoid culture from ATCC R&D scientists. Again, I want to note that from each product page, you can find a direct link to the HCMI searchable catalog in Genomic Data Commons page. Here I want to go into more detail on what ATCC offers in the way of culture support. The basic protocols used for subculturing, thawing, and freezing are largely constant for most organoid models. They really don't differ from one model to the next. The biggest difference between each will be the exact media formulations. There are 8 different formulations that cover the current organoid models offered. They all use a base medium with standard components, but they differ in that most use some kind of conditioned media and different assortments of recombinant proteins and small molecules. On that note, ATCC will begin to offer growth kits to support each formulations. These kits consist of the small molecules and recombinant proteins that I just mentioned, and by packaging them into one kit, it greatly simplifies putting together the complete growth media. These kits will be available in the upcoming months. Now we're moments away from the video everyone is waiting for, and again, this will visually give you step-by-step instructions on how to make organoid cell culture as easy as possible. The video will feature Dr. James Clinton and biologist Himanshi Desai.
ATCC will now be offering organoid growth kits to be used to make complete organoid growth media. The growth kits will avoid the need to make small volume components from larger bulk volumes of growth factors, chemicals, and recombinant proteins. The kits are easy to use, packaged to go immediately in a specified volume of media. Single-use kits avoid freeze-thaw issues that occur when making homemade aliquots, and this ensures consistent performance each time. The kits will be accompanied with online protocols that inform you of which kit to use and how to make the final media. Lastly, ATCC has been using these packaged kits during the manufacturing of all of our organoid models, thus demonstrating how well they work.
And before we start with the video, I want to give an overview of where we are today with the HCMI models. We have launched a total of 150 models, including 80 that are organoids. These include various stages of disease that cover a variety of tissue types. We are planning to launch over 30 more models, at least over the next several months, so stay tuned for that. And now, without further ado, we'll roll with the video presentation and enjoy.
Organoid culture is a three-dimensional culture technique that combines the use of a supportive extracellular scaffold and specific growth factors to generate self-organizing microtissues from normal and diseased epithelial cells that can serve as useful preclinical in vitro models. This video outlines the critical steps involved with thawing, expanding, and cryopreserving human cancer organoids from ATCC. Please note that you should refer to the organoid model specific documentation and written protocols on the ATCC website for additional details. Place a vial of cell basement membrane at 2 to 8 degrees centigrade to thaw overnight. Aliquot into smaller single-use volumes and store at -20 degrees C. Never thaw or store cell basement membrane at ambient temperatures or at 37 degrees centigrade, and avoid multiple freeze-thaw cycles. Single-use aliquots can typically be thawed at 2 to 8 degrees C or on ice in approximately 2 hours. Prepare a 1000x solution of ROCK inhibitor by adding 3 mL of sterile water to the 10 mg vial and mix by pipetting. Aliquot into working volumes and store at -20 degrees C. Avoid repeated freeze and thaw cycles. Prepare the appropriate complete media formulation for the specific organoid model you intend to thaw. Refer to the product literature on the ATCC website for the recipe. On the day of culture, remove the previously prepared complete culture media and place at ambient temperature to warm. Depending on the volume you are warming, this may take several hours. We recommend you only warm the amount required, which is approximately 20 mils per cryovial being thawed. Place a frozen aliquot of ROCK inhibitor and cell basement membrane at 2 to 8 degrees centigrade until completely thawed. Once thawed, cell basement membrane should always be stored at 2 to 8 degrees centigrade or on ice, otherwise it will solidify if the temperature increases. Place a 6-well tissue culture plate inside a 37 degree centigrade cell culture incubator to warm for at least an hour. This step is critical to successful organoid plating. By having the plate pre-warmed to 37 degrees, solidification of the cell basement membrane will occur more quickly and easily. Refer to the lot-specific certificate of analysis on the organoid model for the expected cell count. You will need approximately 100 microliters of thawed cell basement membrane for each 350 to 500,000 viable cells contained within the vial. Cryopreserved ATCC organoids are typically frozen at a density of 1 to 3 million viable cells, so you may need up to 600 microliters of thawed cell basement membrane. Once at ambient temperature, working within a BSC, transfer 10 mils of complete media to a sterile 15 mil conical tube. Remove the cryovial of organoids from liquid nitrogen vapor phase storage and immediately place into a 37 degree water bath to thaw rapidly. Ensure that the neck of the vial is not submerged. Thawing should take under 2 minutes. Once thawed, decontaminate with 70% alcohol and place within a BSC. Using a 1 ml pipette, aspirate the entire contents of the cryovial and transfer dropwise to the previously prepared conical tube of growth media. Centrifuge the conical tube with the cell suspension at 300 times G for 5 minutes. After the spin is complete, transfer to a BSC. Without disturbing the cell pellet, carefully aspirate as much of the supernatant as possible and discard. Resuspend the cell pellet in the appropriate volume of thawed, cold cell basement membrane. Pipette up and down 20 times with a P200 pipetter to ensure the cells are mixed with the cell basement membrane. Avoid introducing bubbles to the suspension and work quickly as the cell basement membrane will begin to solidify as the temperature increases. Remove the 6-well tissue culture plate from the incubator and place in the BSC. Using a P200 pipette, aspirate the cell suspension and dispense as small droplets within each well of the plate. Aim for 100 microliters total within each well, with roughly 8 to 12 individual small droplets. Repeat until all of the cell suspension has been seated. Cover the plate with the lid and flip upside down. While still upside down, put the plate back into the incubator. Flipping the plate can help ensure cells or fragments do not sink to the bottom while waiting for the cell basement membrane to solidify. Wait 20 minutes for the cell basement membrane to solidify. After approximately 20 minutes, add 2 mL of media that has been supplemented with ROCK inhibitor to each well. Dispense the media carefully, such as along the wall of the well, to avoid dislodging or damaging the domes. Return the plate to the incubator. Perform a complete media change every 2 to 3 days. Typically, supplementation with ROCK inhibitor is not needed during routine media changes. Periodically monitor organoid growth and expansion with an inverted microscope with bright field illumination. You should expect fragments to enlarge and form organoids within 3 to 7 days. Prepare for passaging by placing a 6-well tissue culture plate inside a 37-degree cell culture incubator to warm for at least an hour. Ensure you have cell basement membrane completely thawed. You'll need both cold and ambient temperature growth media. Remove your plate of organoids from the incubator and transfer them to a BSC. Carefully aspirate and discard the supernatant while avoiding the cell basement membrane domes. Scrape the domes with a P1000 pipette tip or a cell scraper to remove them from the plate surface, and then transfer them to a conical tube. It may be helpful to add a small volume of cold growth media to facilitate the transfer. With a P1000 pipette tip, pipette up and down approximately 20 to 30 times to break up the domes. Fill the tube to max volume with cold growth medium. Centrifuge the tube for 5 minutes at 300 times G at 4 degrees C. Carefully aspirate the supernatant, avoiding the cell pellet. Resuspend the pellet in dissociation buffer and place in a water bath set to 37 degrees C. Incubate for 5 to 15 minutes or until 80% of the organoids have been reduced to single cells. Monitor dissociation progress with an inverted microscope. Once dissociation is complete, fill the tube to max volume with cold growth medium. Centrifuge the tube for 5 minutes at 300 times G at 4 degrees C. Resuspend the pellet in cold cell basement membrane and pipette up and down 20 to 30 times to mix. Avoid introducing bubbles to the suspension and work quickly, as the cell basement membrane will begin to solidify as the temperature increases. With a P200 pipetter, aspirate 100 microliters of the cell basement membrane cell suspension and dispense as small droplets onto the surface of a pre-warmed 6-well tissue culture plate. You should end up with 8 to 12 individual small droplets. Repeat for all the remaining cell basement membrane cell suspension, dispensing 100 microliters per well of a 6-well plate. Cover the plate with the lid and invert, and place in a 37 degrees C humid cell culture incubator for approximately 20 minutes to allow the domes to solidify. After the domes have solidified, add 2 mL per well of ambient temperature growth media that has been supplemented with ROCK inhibitor. Perform media changes every 2 to 3 days with growth media and monitor growth with an inverted microscope. We typically recommend cryopreserving organoids within 2 days of when you would normally passage them. Aspirate media from wells containing organoids to be cryopreserved. Scrape the domes with a P1000 pipette tip or cell scraper to remove them from the plate surface and transfer them to a conical tube. It may be helpful to add a small volume of cold growth media to facilitate the transfer. With a P1000 pipette tip, pipette up and down approximately 20 to 30 times to break up the domes. Fill the tube to max volume with cold growth medium. Centrifuge the tube for 5 minutes at 300 times G at 4 degrees C. Carefully aspirate the supernatant, avoiding the cell pellet. Resuspend the cell pellet in cold cryopreservation media. We recommend using 1 ml of cryopreservation media per well of a 6-well plate being frozen. For example, if you're freezing 3 wells, resuspend the pellet in 3 mL of freezing media. Transfer 1 ml of the suspension per cryovial. Place the vials in a controlled rate freezing container and store at -80 degrees C for 24 hours. After 24 hours, transfer the vials to liquid nitrogen vapor phase for long-term storage. We just presented the critical steps involved with thawing, expanding, and cryopreserving human cancer organoids from ATCC. As always, careful preparation and following the more detailed protocols available from ATCC will help ensure your success in culturing this advanced in vitro model. For more information on culturing organoids, you can download the ATCC Organoid Culture Guide at atcc.org/organoids.
This concludes our presentation on HCMI organoid models and culture. So to recap, support for these exists in the way of information support on clinical and genomic data via NCI's HCMI searchable catalog and GDC data portal, all protocols and organoid culture media formulations, and coming soon we'll offer core kits to make organoid culture easier. ATCC currently offers 150 HCMI models with more coming in the upcoming months. For more information, please check the ATCC website to download the Organoid Cell Culture Guide, and feel free to watch the video again at your leisure. And thank you to Dr. Shapiro for setting up this webinar. We can now go to the Q&A portion of this presentation.
All right, well, thank you, James and Steve, for that excellent presentation. In just a few moments, we'll begin our Q&A session. Please use the chat function available through the webinar program to submit your questions, and this session will be documented along with the recorded webinar presentation on the ATCC website at www.atcc.org. So we've had quite a few questions come in already. Let's start with our first one. And this one can go to either of our speakers. Are the HCMI models available via any other distributors?
The HCMI, this is Steve. The HCMI models are only available through ATCC. We're the sole distributor for HCMI organoid and even the non-organoid models that are offered.
Okay, good, good.
So we'll add to that. You can certainly buy from an ATCC distributor. Yes.
Okay, so you mean our
overseas
international distribution
Correct.
Partners? Gotcha.
Yep.
Okay. Now maybe this next one's a good one for James. How can someone access the characterization data, and what data is available?
Sure, that's a great question. So there's a variety of data available, and it depends to some extent on the specific model. As a rule, there's whole genome, whole exome, and RNA-seq. Typically, the whole genome and whole exome is from the parental tumor, the expanded model, and also normal tissue. There's a few different ways to access this data. One way is going via the HCMI searchable catalog, which is a resource that is linked via the ATCC website. So if you go to a model's product page, we link out to that. The main source of data is actually on the Genomic Data Commons data portal, which is a website run by the National Cancer Institute. That's where you can find the raw data. Some data is controlled. It requires dbGaP access. This is to protect essentially patients' privacy. You need to provide sort of a study justification about why you want the raw data. Otherwise, they make available mass data that shows a lot of the somatic mutations, and it's very easy to browse and search for what you are looking for there.
All right, good answer. That all makes sense. So I guess maybe this next one's a good one for Steve. Were the organoids generated from cell lines?
No, they were generated from patient directly from patient biopsies. So we receive a biopsy, or the model developers receive a biopsy, and they generate the organoids and send us for distribution. So they're considered more primarily derived from biopsies.
Okay. I guess there's a little bit of an extension from that. So are they generated from purified populations, do you know?
Purified populations.
Or maybe James could take this.
Yeah, so the answer is no. They're not generated from purified populations of cells. There's no selection going on. They are taken directly from, as Steve mentioned, some form of biopsy, be it a needle biopsy from a primary tumor, maybe a literal patient, a piece of a tumor that's been physically removed. There's no purification. It's a heterogeneous population of cells that go into culture and end up generating these organoids. So there's no purification step involved, really.
Okay. Now, are there any restrictions on how the models can be used?
The only not necessarily restrictions, but if you are a commercial entity and you are using for a commercial use, for organoids you have to go through the hub to look into getting a license, and what the license requirements are largely up to the hub. But if you are using for commercial use, there is that you have to look into that. You do not have to have a commercial license to buy the models, but how you use them will determine whether there is a license requirement. But that is for commercial use only. Academic, government labs, this should not be required.
All right, thanks, Steve. Okay, we've had a bunch of questions just come in, but this next one, it's around using the Matrigel or any other related gel matrix product. So the Matrigel seems to dissolve a lot for this asker during the culture. Does anyone else have these difficulties, and what really, I guess, could be happening here? And maybe this is a good question for James to address.
Sure. So this kind of sounds like a troubleshooting technical support type question, which makes it a little tricky without other information. So what I will say is ATCC, we do not use Matrigel. We use a similar product called cell basement membrane, which we distribute. We've used it for all the primary tissue organoids that we've distributed and that we make routinely. I would say if this is happening, I think there can be different reasons. I would say to look at what the protein concentration is of your particular lot of, ECM, whatever it might be. There can be some lot-to-lot variability. We target for the HCMI organoids, usually 12 mg per mil at minimum to embed organoids in. That tends to form it polymerizes really well, and the organoids grow in that quite well. There might be some range in there. Another thing that so I would look at that first, make sure you're hitting some minimum consistent protein concentration. Definitely not. I would not recommend going below like 10 mg per mil. Something else that can happen that can cause the kind of ECM to dissolve, or maybe I would say disintegrate, can be if the density of organoids is really high. If you're seeing a lot of expansion and there's just a lot of organoids within one of these ECM domes, I think it can cause the dome to sort of lose integrity and fall apart. Other reasons that you might see domes falling apart or dissolving, I would look at contamination if that's possible. That can certainly impact the ECM integrity. Of course, you also see that impacting, the organoid culture itself and maybe media changes and things like that. So I would say I have seen that happening before, but it should not be happening routinely. If it's happening routinely, I think you should take a look at your culture protocol. And if you're using, of course, HCMI organoids from ATCC, I encourage you to reach out to ATCC technical support for assistance.
Nice. That was a very complete answer there, James. Okay, for this next question, and of course, if we can't talk about this, that's perfectly fine. But do we have any prostate organoids in the pipeline, and are we expecting to roll them out? And this can go to maybe well, either of you guys can handle this, I'm sure.
Yeah, I could just start by saying, yeah, we definitely want some prostate models. We don't have any yet. It's kind of we would have hoped that we would have some by now. I don't know if James, if you've seen any difficulty with prostate models, or it's largely just we haven't really received a lot from the developers yet.
I would agree that we have not received a large number of prostate organoids yet. They have not been a focus, I think, of the HCMI previously. That's not to say we might not receive them in the future. I could not give you a timeline for when we might release prostate organoids.
Right. I mean, yeah, I'll just say we're going to continue to launch models, a few per month for ongoing months from now, with no real. We keep extending it more and more, longer and longer. But yeah, we definitely will continue to launch more models, and we hope to see some prostates come in over the next few to several months.
Okay. Now, this next question that's come in is very technical in nature, so I think this will go James' way. For washing and resuspending organoids, do we need to use medium with factors like the arspondin or noggin or EPF?
Yeah, that's a good question. I would say you don't need to. You might want to. Typically, when you're handling organoids, especially if you're dissociating them, if you're collecting them off the plate, washing them, dissociating them, resuspending them, that can be stressful for the cells involved. Keeping them in a complete formulation with the various growth factors, the sources of Wnt, arspondin, noggin EGF, and the small molecules, I think can be helpful. Sometimes also including ROCK inhibitor can be helpful. That being said, not always do we necessarily do that, and you can use more of a basal media. I would still include something like some of the supplements, like B27, glutamine, heat, some buffering in there. Since, again, if you're doing something like passaging and they're incubating in a dissociation buffer afterwards or before, that can be stressful. So I would suggest doing it. You need to do it. Not necessarily, but that it might also vary depending on what type of organoid you're working with, what specific donor you're working with. They can behave a little bit differently and be more or less sensitive to the stress of being washed and dissociated and resuspended and passive.
Okay. Now this next question is pretty general, but could either of you comment on what kind of assays you could use the organoids with?
Sure. So organoids are amenable to a lot of standard lab assays that you might do with, a continuous cancer cell line. We routinely do a lot of ICT with them to look for, expression of, protein markers, various protein markers using immunofluorescence. You can do various viability assays. You can do flow cytometry with them, another form of immunocytochemistry. You can do drug assays. You can plate them into multi-well plates. You can treat them with drugs and then do a viability assay, something like ATP-based. We do that routinely. You can grow organoids, treat them, and then collect nucleic acids for downstream something PCR-based. You can collect protein for Western blots. basically anything you can do with, I would say most in vitro models, be it, again, a typical cancer cell line, you can probably do it with organoids as well. The protocols might be a little bit different. Certainly you have to contend with the presence of ECM, where they've been propagated in, and the fact that they're much larger multicellular structures. So maybe you need to reduce them to a single cell first before you do downstream processing. But I would say almost anything you can do with a cancer cell line, a 2D adherent cancer cell line, you can do with an organoid as well, the same assays.
Okay. Now, have we tried to make any organoid models from PDX or CDX tumors? Is this something that we would think of doing?
So I'll take this one. So, the answer is no. Intentionally, these models are directly coming from patients into culture. So, PDX models are not part of or outside the scope of what the HCMI is looking to do. So, that being said, could it be used with our organoid kit with these media formulation support, a PDX-derived or other types of organoids from other tissues, other donors, other cancer types? The answer is maybe, but we have not investigated that.
Okay, yep, that makes sense. Now the next few questions are just kind of around what we have available, and so I'm just going to name out the tissue types. So we have people asking about ovarian organoids, lung organoids, and bladder cancer organoids.
Yeah, I can. Okay,
go ahead, Steve.
I was just going to briefly say what. So you said lung. We have about 4 lung models. One's organoid. The others are sort of mixed adherent, 2D adherent. But one PDM3 is a lung organoid. What else did you say?
Ovarian.
Yeah.
Yep. And then the other is bladder.
Yeah. We do not have any ovarian ones at this point. We have gallbladder models, and of those we have just about one organoid model for that, which is PDM 273. Yeah.
How about liver? Since we're just naming what we have.
Right. We have one liver model as well.
Popular, I would think.
Right. Mm-hmm. A liver model as well, organoid, PDM9.
And some biliary tract, maybe specific tissues associated with the biliary tract, like ampulla vator.
Specifically ampulla vator, yes, we have some models, 3 of those. Organoid. Yep. Mm-hmm. And Brian, I believe we have, and I think we're updating it continuously, a brochure that can be available that has a list of the models by tissue type, correct?
Correct. And that's going to be live on the website, the updated one shortly. And, if you just want to look at, what organoid models we have, you go to www.atcc.org/organoids, which is here on this slide. If you want to see all of the HCMI models that we have overall, you can go to www.atcc.org/HCMI. So yeah. Okay, we've got one more question that's come in. So I'm going to basically call last call for questions as I ask this. And this is also a very technical question. So James, does cryopreservation reduce the initial growth of the organoid?
Does cryopreservation, so is this, I assume this question is kind of referring to maybe recovery from cryopreservation. So if you're starting with a cryopreserved amp of organoids, you're thawing it, does it reduce the initial growth? I would say, again, this is something that can vary depending on the specific model. There may be some lag time, but typically robust models will recover from cryopreservation just fine and will expand well. Again, it's going to depend heavily. So, just as a reminder, every individual organoid model is derived from, for the most part, a different patient, a different tumor, a different cancer, and there is a lot of variation inherent to that. So the models can behave differently. That being said, I would say maybe, yeah, in the initial recovery from cryopreservation, they might grow a little bit slower than they will maybe on that after that first passage, but not dramatically slower. And I wouldn't say that's necessarily something that even occurs for every model. Some models like colon tend to be incredibly robust, grow very quickly, and they will recover from cryopreservation very easily. Other models, particularly I would say some metastatic models, memory models, grow slower fundamentally, and they may also have a more difficult time expanding immediately following thaw from cryopreservation.
Okay. No, that's good. Good answer. Good answer. Now, another question has come in. Since the organoids are heterogeneous, bulk RNA-seq may be difficult to interpret. Has any single-cell characterization been done?
Sure, I can answer this question. Yeah, no, it's a very good point. And there is an intent to do single-cell seq. It's not something single-cell RNA-seq. It's not something ATCC has done. I believe some of the model developers are interested in looking at that. Actually, this is something that has been discussed at ATCC as well. But yeah, I agree with you. I don't have that data to share with you. I don't know when it will be available. I do believe, though, it will be done for at least some of the models at some point. So not a great answer, but it's the reality of the nature of the model because they are heterogeneous.
Okay. And so two more questions have come in. This next one's kind of connected to the one that I just asked you. Do you know if the heterogeneity of the organoid population remains constant? Or I guess, you think of like all the different cells that are in the colon, all the different types of cells. If you're making gut organoids, do they remain the same like over passage?
Again, yeah, it's a great question, and it's a question mark. Like, does it? And it depends. First of all, on how what is your definition of heterogeneous? What is by what measure are you making that assessment? I think we ultimately really don't know. Over what time frame? We haven't. These models, to a large extent, haven't existed that long. Most of them were actually only derived in the last couple of years. No one has taken them out 50, passages to see what they look like at that point. As part of this project, models are being resequenced to look at least at the genotypes to say, are certain key mutations that were present in the tumor in the original model still present in the expanded model? That's sort of the best. That's really the only data we're getting on that, or that we have right now. No one can promise you that any individual model will remain, whether the genotype or the phenotype will remain perfectly consistent or perfectly heterogeneous in the same sense over the course of culture at this time. It's an interesting scientific question, I think, that the HCMI models are kind of well positioned to answer. Ultimately, though, it's not one that we have an answer right now.
Okay. And for this final question, are multiple models from the same patient available?
Yes. Oh, go ahead. I was just going to say, yeah, we have a few models that have a few models that have a corresponding model from the same patient. So so it'd be the type where you have like the primary and then the metastatic tumor and so forth. You can, on our website, you can, well, not that easy to find. The website has, on the product page, if the model has a donor, it will be listed there. But usually, if an ID3 number ends in a additional letter combination, like 06A, for instance, that means it has another model corresponding with. Like for instance, if you see something that says HCM-CS0089-C25-06A, it'll have a corresponding part that's HCM-CS-SHL-0089-C25 without that last little letter component there. But that information can be easily made available if needed. If anyone were to contact tech support and ask that, we can probably give you a brief summary of them.
Right, and kind of on top of that, that information is also in the comment section in that product detail page. So, all right, well, at this time, we'll conclude our Q&A session. So thanks again to James and Steve for the excellent presentation and doing a great job fielding the questions. And thank you everyone for attending the webinar. For more information about culturing organoids, please navigate to www.atcc.org. And thank you again for your time today. Have a great day.